Hi, my name is Akash Fadlam, and my project is the urolithin A analog ASR600 inhibits the growth of muscle-invasive bladder cancer in the G2M phase. So bladder cancer is one of the most common types of cancer in the world. In 2019, it caused around 17,770 deaths. And there are two different types of bladder cancer, non-muscle invasive and muscle invasive. Non-muscle invasive is less dangerous and more treatable. However, muscle invasive bladder cancer is very dangerous as it is able to spread to other organs and has a higher likelihood of death. So the two bladder cancer cell lines that I use are both muscle invasive, and they are TCC-SUP and UM-UC3. And also in the cell cycle, there are four phases: the G1 phase, synthesis phase, G2 phase, and mitosis, also known as the M phase. So 2 years ago, I performed an experiment where I tested the effect of urolithin A, which is a molecule found in pomegranates, on bladder cancer cells. And I found that the urolithin A successfully inhibited the bladder cancer cells. However, the concentration that was required to do so was very high. So last year, I attempted to improve on that. I used ASR 600, which is a structurally similar molecule to urolithin A. And I used it to test on bladder cancer cells, and I found that the IC50, which is the concentration required to kill 50% of the cells, was greatly decreased from when using urolithin A. For urolithin A, the IC50 was 15 micromoles. However, when I used ASR 600, the IC50 was only 3.12 micromoles. So this year, I wanted to see in which phase of the cell cycle was the ASR 600 able to inhibit the bladder cancer cells. So my hypothesis is, if ASR 600 is used to treat the bladder cancer cells, then the cells will undergo apoptosis in the G2M phase of the cell cycle. So in order to test this, I used a flow cytometry assay. And this assay is able to calculate the percentages of cells, the percent of cells in each phase of the cell cycle. So first, the cells were seated in 6 well plates and treated with ASR 600 for 24, 48, and 72 hours. The cells were then washed with PBS and fixed with ethyl alcohol at 4 degrees Celsius overnight. Then they were re-washed with PBS the next day and incubated with RNase A for 30 minutes, which was followed by a staining of propidium iodide for 30 minutes. The staining of propidium iodide allows the flow cytometry machine to detect the cells. So the results of this showed that during the G2M phase, there was a discrepancy between the control and the treatment cells. There were significantly more cells in the G2M phase in almost each time point in the two different cell lines, with the highest discrepancy being around 40%. This means that the cells underwent apoptosis in this stage, as the assay also includes dead cells in its calculations. This means that there was an accumulation of dead cells in the G2M phase. After this, I did a Western blot assay in order to confirm that apoptosis was occurring in this phase. I used Western blotting to detect proteins that are associated with the G2M phase. These proteins are cyclin B1, PCDC25C, CDC25C, CDC2, and PLK1. So the results showed that in cyclin B1, CDC25C, CDC2, and PLK1, there was a decrease in expression. Now this is the expected result, as those proteins are all G2M markers. So that means that if there is bladder cancer cells going through the G2M phase, they will have high expression. And as the bladder cancer cells are being inhibited in this phase, the expression will decrease with the treatment of ASR 600. So this confirms that apoptosis of the cells occurs in the G2M phase. So further, the PCDC25C is the unactivated version of the CDC25C. So as the CDC25C decreases in expression, there's a lesser need to activate PCDC25C and make CDC25C. That's why there's an increase of expression of the PCDC25C. And this occurs in both cell lines. So here's a simple diagram to simplify my project. So ASR 600, which is a structurally similar molecule to urolithin A, which is found in pomegranates, treats the bladder cancer cell, and those cells go into the cell cycle. There is then growth arrest in the G2M phase, which leads to inhibition of the cell cycle. So in conclusion, the data received confirmed the hypothesis, as I predicted that the cancer cells would undergo apoptosis in the G2M phase, and according to the flow cytometry assay, they did. It was also found that G2M checkpoint markers also showed a decrease in expression, which further confirms that the apoptosis of the cancer cells did occur in the G2M phase. In practical terms, this means that ASR 600 can become an important molecule in the finding of a more efficient cancer treatment option. Thank you.